me interested in, in biologics in general. There's so many different varieties of biologics that I just keep learning. And I think that's been awesome. So where I'm working right now is a company called Prothelia. It's a startup company. We have 9 people total. And what we're doing is we're developing a first-in-class protein replacement therapy for a congenital muscular dystrophy. So these kids, I'll go through the progression of the disease, but these kids basically know that there's something not quite right with them at birth. The disease that we're working on is called LAMA2 congenital muscular dystrophy. It's a rare form of muscular dystrophy, and our estimated patient population is about 8,000 worldwide. So definitely considered a rare disease. So, so these kids that are born with LAMA2 deficiency, straight away at birth, you notice a lot of muscle weakness. So they call it floppy baby syndrome. Basically, around a couple months old, kids can usually hold their head up or at least start moving their head around. These kids, you just don't see that. Then there's, progressive motor milestone delays. So crawling, rolling over, that sort of thing just doesn't happen. And these kids actually never are able to walk. So they're in a wheelchair for their whole lives. Around 5 years old, they start to develop, or 2 to 5 years old, actually, they start to develop these things called contractures. And so that's when their joints start kind of getting locked in and turned in. So they don't have the flexibility anymore. They're just almost like stuck just because they don't have the muscle support to move around the joints and the bones. Around 6 to 8 years old, they start developing scoliosis, and that requires surgery as early as 6 or 7 years old, actually, in some cases. And then at about 11 years old and in early teens, they start to have issues with feeding and breathing. And so they end up going on G-tubes and needing respirators around 11, 12 years of age. So, it's not, these kids do survive into their 20s, but they need serious interventions pretty early on in their life. And as of right now, there's no commercially available treatments for LAMA2 CMD. So our approach to LAMA2 CMD is, like I said, a protein replacement therapy. So in normal muscle, which is shown over here on the left, there's a laminin, there's a protein called laminin 211. And so this laminin binds to the surface of the muscle cells through receptors. And then it forms this layer or network of other laminin molecules by basically linking with other laminins across the surface of the muscle. And so laminin is the first component in forming the basal membrane layer on muscle cells. And then there's other molecules that kind of pile on top, like collagen and stuff like that, to make the whole muscle matrix. For kids that have this LAMA2 mutation, what happens is they're not able to express the laminin 211. And as a result, they can't form this nice network on the muscle cells. In addition, what's upregulated is this laminin 411, and that laminin 411 is kind of like a defective molecule. So it's not 100% defective. It can sometimes bind to the surface of the muscle cells, but it cannot form a network. It's missing this head group here. So it can't bind to other laminins and make that nice lattice on top of the muscle. So then what happens, you can't form this basal membrane network, and then the muscles start rubbing on top of each other. There's no kind of like lubrication between the muscle cells, and they eventually undergo apoptosis, and then you see all of the effects that I just talked about on the last slide. So our approach is to replace laminin 211 with another laminin that's a perfect analog of laminin 211. It's called laminin 111. So the only difference is this, there's 3 subunits that make up laminin. It's called alpha, beta, and gamma. So the only difference between laminin 211 and 111 is this alpha unit, which is shown here. So now our new one, our alpha 1, is in green as opposed to the last slide where I had the lamma 2 depicted in pink. And so what our hope is that when we deliver LAM 111, we can restore the basal membrane lattice that laminin makes, including probably some of the mostly defective 411 and whatever 211 is around. And that will rescue the muscle, and at least we don't know if it would reverse, the disease, but at least we hope that it would stop the progression of the disease when we start administering it. So happy to, like I said, any of these topics I can do a whole hour on. So I'm happy to answer any questions on the disease if you're curious. I guess we'll save that to the end of the talk. So now kind of more broadly, I want to go over what my role is so far in my career, and especially at Prothelia. So what are biologics? A biological product consists of sugars, proteins, nucleic acids, any combination of which I worked on before, could be living things such as cells, like you probably have heard of cell therapy, virus, there's a virus delivery of DNA, that sort of thing. And the key is that they're produced or obtained from a living system or from recombinant DNA technology. And, as you saw, all the different biologics that I've worked on, there's many categories. There's old school, like blood products, like albumin. There's proteins, vaccines, viral vaccines, tissues, genes, all sorts of stuff like that. And what's cool is that, every year we're seeing more and more biologics, both classes of biologics and just numbers of biologics that are making it to the market. So I just have 2 examples that are really common right now. This is a monoclonal antibody, which we'll talk about more as kind of a case study for process development and manufacturing. And this is a viral vector called adeno-associated virus that's used to deliver DNA to people. So I like this image. I did not make it, I stole it, but it shows you just how complex biologics are. So, here's our small molecule drug. This is aspirin here. This is actually not even drawn to scale, right? It's 180 Daltons. And then you get to the biologics, and insulin's pretty small, 5,700 Daltons. And then you get bigger and bigger. So an example is human growth hormone, which is about 22 kilodaltons, monoclonal antibodies, 150 kilodaltons. And then the AAV that I showed you on the last slide, that's about a megadalton. So getting very big, very complex. So there's benefits and drawbacks to small molecules versus biologics. Small molecules are definitely easier to manufacture, lower molecular weight. Everything's very controlled. It's all chemistry. Whereas biological products, we're relying on living things to make our drugs for us. Small molecules are much more easily characterized with analytical methods. Biologics, not so much, because you can have quite an array of variants that you're expressing just because of the nature of how you're making your product. The structure, you may not really have a good handle on the structure, but we're getting better at coming up with tools to measure that. Small molecules are not usually immunogenic. So, the immune system doesn't see it as being foreign, whereas biological products, because they're bigger, often are immunogenic. And so that is a concern that we have to pay attention to. Small molecules are more toxic and do have more off-target effects. So that's definitely where the biologics are definitely a lot better. And just because of the lower toxicity and the higher specificity to the target. So as I mentioned, biologics are kind of, starting to make up more ground in the overall landscape of the drugs that are approved and out there. The drawback right now, and it is getting better, but that biologics are a whole lot more expensive to make. They take a lot longer to make. The way you make them is much more complicated. And so it takes about $1 billion. These numbers change, depending on what we're looking at, but about $1 billion to take a drug from discovery to approval and getting out there on the market. And you can see, so the R&D expenditures, which is shown in this like red line with the blue, early in the 60s, there were no biologics. It was all small molecules. The amount of money being spent on R&D was very low, but as biologics became more and more prevalent, those R&D costs go up quite a bit, and hence why we see drug prices go up so high. We have to pay for the R&D costs, and they're just inherently more expensive. But that's the cool part for people like me that are process development scientists or manufacturing scientists, that sort of thing, because that's the innovation in that area is the big draw. Innovation in kind of in the manufacturing area is what's going to drive these drug costs down. So there'll always be a place for innovation in biomanufacturing. So like I mentioned, I'm going to kind of walk you through how we develop a process for making a biologic, and we'll use an example of monoclonal antibodies. Those are kind of like the benchmark right now in biologics for manufacturing. So I'm going to give you a one-slide overview of drug discovery. I mentioned to the class earlier that if you want something more in depth, this is exactly what my husband does. So he could come and give you a talk on this. So I'm going to kind of give you the high level and gloss over it a little bit. But basically, the approach, you got to figure out what disease area you want to be. And so sometimes companies choose, just based on their expertise at their company, they'll say, okay, we're just going to work on diseases in the central nervous system, or we're just going to work on, oncology or that sort of thing. So you decide your high level disease area. And then, when you're choosing which actual disease to treat, you need to really make sure you understand all of the aspects of the disease, because before you even start investing a ton of money in this, and this could be as simple as reading a bunch of papers in that area, not even doing experiments. And then from there, you start designing, figuring out like which molecules are going to be most effective for that disease. It could be small molecules, it could be biologics, it could be DNA, it could be protein, right? So it all just kind of depends on what sort of diseases you want to treat. So then once you decide that, this is an example for monoclonal antibodies. To make your antibodies, you take your antigen, which is whatever molecule you want your antibody to bind to, and I'll show you how that all works in the next slide. So say you want to develop like the COVID antibody, right? So you'd take the spike protein, you'd immunize mice with that, you isolate the immune cells from the mice, and you end up with these antibody forming cells. And then you fuse those with tumor cells in forming these cells called hybridomas that basically display the antibody on the surface. And then you use cell sorting to screen your hybridomas to separate out the cells that are producing the antibodies that bind to your target. And then you then sequence the antibodies that are being produced, and then you start producing those antibodies, and then you want to eventually screen those for a variety of things, including efficacy. And then the other thing that's important to look at is manufacturability. So, it's great if you can make an antibody that binds to your target molecule, but if it like completely falls apart at room temperature because it's unstable, then you're dead in the water. You don't have a drug to develop. So all of these things happen pretty early on in the drug development timeline. And can ultimately save you a lot of money down the road if you catch these things early on. So just quickly, I wanted to show you, because we're going to talk about a lot about monoclonal antibodies. So I wanted to make sure everyone's on the same page. So their MABs are probably the most abundant and well-characterized biologic on the market. This is the structure over here. So there's, it's made of a heavy chain, 2 heavy chains and 2 light chains, they're called. So these are the heavy chains in blue. In the middle, the longer chains, and then the light chains are shown here on the outside. There's 2 regions of the antibody, without going into like major detail. There's a constant region, which is shown here, so that really doesn't change, especially if you're developing a bunch of your company is developing a bunch of antibodies as therapeutics. You pretty much keep this portion of the molecule the same. And what you want to change is this variable region, which is shown here in the VH being variable region for the heavy chain and VL being variable region for the light chain. And the reason that those change is because that's actually the portion of the molecule that binds to your antigen or your target molecule. So in this case, this is a cancer. You're targeting some sort of cancer molecule. You want it to bind really tightly to the antigen binding site on your MAB, so then it can be, for instance, cleared from the body. What's also pretty interesting in the kind of the evolution of monoclonal antibodies since basically like probably the 80s or early 90s, is early on we were just making mouse antibodies. So we would isolate the antibodies directly from these mice, and they would have, basically mouse characteristics. And so we didn't have a good enough technology to change the amino acid sequence to make it less like a mouse antibody, more like a human antibody. So those were pretty immunogenic. Your body's going to see those as being foreign. But over time, as we understood the technology better, we started making chimeras. So a mixture, so like the variable regions would be mouse, and then the constant region would be the human. And then eventually we figured out how to humanize. So basically how to change the sequence of the antibodies to look less like mouse and more like humans. And now what's available on the market is these mice that have human immune systems. So when you immunize your mice to pull out your antibodies, those are it's a human, basically a human immune system that are making these antibodies. So that's kind of how we get to our antibody. And now I want to take you through the various aspects of biomanufacturing. So you can see how we go from basically like the bench to, these or milligram scales to what we need for commercial landscape, which is, kilograms or more. So, yeah, so I just mentioned this. So process development is the group within a biologics biopharma company that take the process from the discovery group, where they're familiar with the molecule, they're able to make it on their own, but it's only for maybe in vivo or in vitro studies, not it's still pretty dirty at that stage, to kilograms or more, where we're getting into making product that's good enough to give to people. So that's clinical manufacturing. And then commercial is when it's actually on the market and you're just pumping out product to sell. So what I like about process development is that we're positioned very central to the organization. So we have to interact with the discovery folks because we need to understand what they learn. And sometimes we have to make material for them to use in their animal studies, stuff like that. We are very intertwined with the manufacturing groups because we need to figure out how to make it. And then they're the ones that make it. So we pass it over the fence to them. We need to be very highly involved with like the quality people. So making sure that we're making good quality product. The regulatory folks need to understand need to make sure that we have a good enough understanding and confidence that our molecule is safe. And that, you know, the business even, right? Because we have to make sure that these drugs that we're developing, like, yes, we always want to help people, but then there's also those stinky business people that want to make sure that we're going to make money off of our product too. So we often are talking to the business side too. So, but the overarching goal when we take projects into process development is safety, efficacy, and consistency. So we want to make sure that we're making a safe product. We want to make sure that the product that we're making, because we're manipulating a lot of things when we're trying to get, you know, pump out lots and lots of protein. We want to make sure that drug is still efficacious and doing what we want it to do. And then we want to make sure that every batch we make is making consistently the same drug. It has the same product profile from batch to batch. So there are three main areas of process development, and people, like, my core competency is purification or downstream process development. That's how I came out of my postdoc. I got a job in purification, and that's what I love. It sounds silly because it's just a small piece of making a protein, but you know, there's a lot of aspects, and you really get drill down into purification and understand it well. There's also cell culture groups, so it's called upstream process development, and those are the folks that, you know, figure out how to make the cell line, so get the DNA into the cells, make sure that we're pumping out as much protein as we can with the process. And then the last step is filling, and so we call that drug product formulation and fill finish development. So I'm going to dive into each of these areas so that you can get a flavor of what the important things are with each. So as we go through the development for all areas of process development, we kind of follow this kind of funnel paradigm. So first, we're doing development at a small scale. So we're doing lots of experiments. We don't really know, you know, what parameters are important. So we're doing all sorts of experiments. A lot of them are going to fail, but that we need to have that sort of failure so that we can understand where we can operate and make the best product possible. And then we say, okay, you know, we think we understand this process well enough. Now we're ready to scale up the process. So let's say we're going from milligrams to 100 grams or something, right? So that's this kind of middle area. So we make sure that that everything we learned at the small scale works at like a moderate scale. And then eventually we reach the stage where we really need to make sure that this process is locked in. We know exactly what's going to happen every time. And if something goes off the rails, we actually have done experiments to kind of to show that the product is safe or not safe, because these batches cost like $10 million. You don't want to be failing a batch. So the more you understand your process, the better. So here's a generic mAb production process. So like I mentioned, mAbs have been made for a long time. We understand a lot about how to make them and how to make them well. So what people generally use a platform approach for monoclonal antibodies. So most of them are made in Chinese hamster ovary cells. These are obviously they're from Chinese hamsters. They're mammalian cells, and what you do is you transfect the cells with your DNA. So this would be our heavy and light chain genes, plasmids with our heavy and light chain genes for the antibody. And eventually we pick one clone off of the plate and we start to scale it up. So we go into a small scale shake flask, usually. This is bioreactor shown here. But then we scale that up to a small bioreactor, and then eventually to a large bioreactor. And so then when you're producing mAbs, as opposed to like when you're producing protein in E. coli, the CHO cells actually excrete the antibody into the media. So instead of lysing the cells like you would with E. coli, in this case, you just need to get rid of the cells. So that's done either by centrifugation or using these filters with really large pores to hold up the cells. And then there's a series, which I'll go into all these steps in detail as we walk through, but there's a series of chromatography steps where we're purifying the antibody, making sure we get rid of all the cell junk and stuff like that. And then eventually we end up with our purified protein. And what's important at the end is we need to make sure that this protein is put into a buffer at a certain concentration that's will preserve the stability of the protein long term, because we don't want to make a batch of protein and have it only be stable for 2 months, because then we would be constantly having to make new batches of protein. We generally want at least 2 years of stability of our protein, just to make sure we can get the material where it needs to be and into the patient. So first we'll start with cell culture. That's the first stage of producing an antibody. So what we do is we take those CHO cells, or it could be any cells. Like right now I'm working on a human cell line, actually, instead of the Chinese hamster cells. And we transfect the cells with the DNA, like I mentioned. And what we do with those transfected cells is we plate them, and we choose one clone. So there's actually these, it's shown very small here, but there are these these robots that will go in, and it'll zoom in on these wells that have a bunch of cells in them and just pick up one. And why that's important is because even though you're transfecting the cells with the same DNA, each of those cells, not each of the cells, but there are populations of the cells that are going to have different phenotypes from each other. So, some will produce more protein, some will need more glucose to grow. So they'll have all sorts of different characteristics that you need to make sure that you understand well. So if I go in and I take a pool of those cells, my ability to predict whether my batches are going to be consistent goes down. But if I go in and I pick one cell and start the cell culture process with one cell, then I can be pretty much assured that I'm going to understand those cells pretty well. So we then pick clones. These are called clones. So we pick clones. And in this case, when we're using robots like this, you can do like 1,000 clones. And you put them into 96 well or 386 well plates, and you start scaling them up. And you, depending on what you want your outcome to be, you may have certain characteristics of your protein that you want to come out. Usually at this stage, you just want to make a lot of protein. So you'll continue to scale up and look at the protein that's being made. So in Shake Flash shown here, there's also these miniature bioreactors that you can use. So that gives you kind of a better prediction of how the cells and how the protein is going to look when you ultimately scale up to larger scale bioreactors. And then eventually you whittle down from 1,000 clones to maybe 2 to 4 clones, and you'll look at them in larger scale bioreactors, maybe 10-liter bioreactors. So this whole process is actually pretty time consuming. It's not a lot of work, but it just takes a long time. So it's about 4 to 12 months, depending on the technology. And in the end, you want to make sure your cells are genetically stable, so you're not losing copies of your genes as your cells grow. And you want to make sure that the product quality, so the protein you're making, isn't impacted. So then once you've picked your clone, then you want to make sure that you can pump out as much protein as possible. But that's tricky because you're taking these Chinese hamster ovary cells, you're putting in a human protein, genes for a human protein, and you're wanting it to pump out a ton of protein. So it's tricky, and there's lots of things which I won't go into in detail, but there's lots of things that we have to pay attention to. So we need to make sure that there's enough nutrients. Sometimes temperature is important. The osmolality is important. All these things are important to make sure that we can have the cells make as much and a good quality protein as possible. And so I thought this image was interesting. So this shows from 1985 when antibodies were first starting to be produced in CHO cells to today, how much we've improved the protein yields just really by understanding the protein and the cells better and better over time. And this comes from research groups at universities. It comes from companies. Lots of people are looking into this. So we're going from like making about 200 mgs per liter. This is in grams per liter. So 200 mgs per liter to now making consistently 3 grams per liter. I've definitely heard instances of greater than 10 grams per liter that people can achieve today. This is all, top secret stuff, but so it's really impressive. I mean, it's not that long of a period of time to increase yields by, 50-fold. And then what's important too, of course, is that if we increase the upstream yields, that's the very first step of our process, we're going to drive down the cost of these expensive therapeutics. And this is just quickly to show. So when we do upstream process development, we start off in those little miniature bioreactors or shake flasks. So these are like 15 milliliters, 250 milliliters, and it's all controlled by robots, which is pretty cool. And then you scale up 50, 200, and even up to 2,000 liters. There's even 20,000 liter bioreactors that you need like ladders and stuff to get on. And so it's important to understand your process, because scaling up processes also poses a lot of challenges. So now we'll get quickly into downstream process development or purification. And so our overarching goal in the purification side of things is purity, safety, and yields. Right? Yields are always going to be important because that's where the money comes from. But purity and safety are important. And so unfortunately, there's always a sacrifice between yield and purity. It would be awesome if you could have a high yield, high purity process, but that's usually not the case. So what we need to worry about in downstream process development are impurities and contaminants. So impurities can be product related. So aggregates where you get multiple protein molecules sticking to each other. You can get fragments. So, maybe from proteases or something like that, you have a piece of your molecule clipped and you don't want to be giving that to patients. There are host cell impurities like host cell proteins. So as the cells definitely aren't just excreting our monoclonal antibody, they're also secreting all sorts of other proteins. So we have to make sure we can purify those away. There's also, the cells don't live the whole time that you're running your upstream culture. So you always have cells that are dying, like they're always multiplying and dying at the same time. So we have host cell proteins just from those dead cells and the pores opening up, the cell membranes were opening up in those cells. And same with DNA. That's a cancer concern, because if we end up delivering DNA into people and that actually gets into the nucleus of the cells, then that could be bad. And then there's also, the cell culture additives and buffers and stuff like that we need to purify away. There's also external concerns. So just from, we manufacture these drugs in clean rooms, but there are cases where there are contaminants that are found. So that could be viruses, it could be bacteria, it could be fungus. These are all under control, but it does happen. So we want to make sure that our process is pretty robust to remove things we know we have, but also things that we could encounter. So here's a generic process for a MAB purification. So this is kind of that platform that I was talking about. So we get the cells we take the bioreactor and we want to clarify the cells away. So we can either use a fancy centrifuge or depth filtration, which is just a large pore filter, like analogous to your 0.2 micron filters that you might use for buffers or stuff like that. It's the same idea. Typically there are three chromatography steps. The nice thing about antibodies is there's a resin that specifically will bind to a portion of the antibodies. So that does a huge amount of work purifying away impurities. And then generally there are two other columns. Generally there's an anion exchange chromatography step, which I'll go into a little bit. And then there's a third step. In this case, it's a hydrophobic interaction. So it takes advantage of hydrophobic patches on your antibody. There are also 2 steps that are built into these processes to remove potential virus contaminants. One, which is not shown here, is there's a low pH inactivation step. So some viruses are sensitive to pH, and but antibodies are pretty robust, so they're not going to be hurt by that low pH. So we can get assurance there that if there's those type of viruses, we can remove them. And then there's also these really small pore size filters that we use called virus filters, and that will retain any contaminant viruses while letting our antibody come through. And then the last step is the final formulation. So we use these membranes that allow us to concentrate and buffer exchange. Like they're kind of like they are the same as the centrifugal filters that some of you guys have used probably to concentrate proteins, but it's just a different format that you use, but same operation. So everything's about speed in drug development. So we have a couple of tools at our disposal to accelerate downstream process development. There's high throughput technology. So they're just like I showed you with the small scale bioreactors. There are also these robotic systems where you can do purifications in plates, 96 well plates, or these little miniature columns. And so that allows you to screen a lot of different conditions all at the same time. There's also these FPLC instruments that you can automate. So you can just keep running back-to-back runs. You don't have to be there watching it. And the other thing that's kind of making a comeback, I guess I would say, is mechanistic modeling. So especially with antibodies, we know so much about them now that people that are good at math and stuff, they can generate these models to predict how the purification processes are going to perform. So here's a quick example of for downstream development, and this is anion exchange purification. So antibodies at like pH 7 under pH neutral conditions are generally negatively charged. So positively charged, generally positively charged. So we choose this anion exchange membrane, which has these positive charge groups on it, so that we can bind negatively charged impurities. Like DNA has that phosphate backbone, so it's negatively charged. Viruses generally have a coat that's negatively charged. So all of those impurities are going to bind, and our protein flows through the column. So the nice thing about that is instead of having really big columns, because we need to bind all of the protein that we're loading on it, we can actually pack really small columns, so that because we're only binding a small percentage of the chemicals or compounds that we have in our antibody solution. So process related impurities are in there, like I mentioned, host cell proteins, DNA. Some product related impurities will bind. So sometimes charge variants will bind. That all depends on how the surface charge on the molecule looks. And also the aggregates also have a different surface charge compared to the singular antibody. And so in the data that I'm going to show you on the next slide, just as an example, this was done with, it's not that important, but just to expose you guys to it, it's this statistical method called design of experiments. And so in this case, we wanted to understand the whole design space of where we might want to operate this anion exchange step. So we did 20 different conditions plus 2 confirmation runs. So just make sure that our small scale model is working well. We do 2 conditions, 1 condition in triplicate, basically. So all in, we did 22 experiments. And the things that we wanted to look at were how much protein we were loading, the pH, and the volume of wash. So I'm just going to show you a couple of examples, not the whole experiment. And the important thing that we wanted to see is yield, which is obviously important. We don't want to lose all our product, and the high molecular weight or aggregates. So we do all these experiments, and then we put it into the software, and we get these graphs called contour plots. And so this is an example of the load pH and how much we're looking at the load and wash pH, and then how much protein we're loading. So, and both, increase across the axis. So we can see that the yield percent, which is shown here in these contours, goes up as we decrease the pH, and goes up as we increase the load. So say we said, okay, we can handle a 90% yield for this process. So then we could operate this process anywhere in this corner of the design space. For high molecular weight removal, it's the opposite. So this is that balance, the yin and the yang, right? So if we operate in that bottom corner, now our high molecular weight removal, our aggregate removal is less. So probably where we are is operating kind of in this happy medium. It all just depends on what you need from your process, but which is determined, basically continues to be determined as you develop your production process. And then I showed you how the upstream process scales. So this is how the downstream process scales. So, I mentioned that there are these 96 well plates you can do experiments in, or these little miniature columns. Then that eventually gets scaled up to these more moderate sized columns. And then eventually when we get into the real manufacturing for the clinic or commercial, we get into really big columns. So this is like, the size of probably like R2-D2 or something like that, like a trash can. So these columns can get pretty big. And then I'll try to go fast. So the last step of making a drug is to put it into the final vial. But that's not as simple as just, squirting into a vial and putting a cap on it. There's a whole lot of stuff that goes into that. So everything up till now, the upstream and the downstream process, that's considered drug substance. And then when we put into a vial and it's ready to go out, that's called drug product. So you'll see that drug product nomenclature through here. So again, goal, just like all the other steps, deliver safe and efficacious drug product to the patient. And then the things that go into that are optimizing the formulation for stability and bioavailability, because you don't want to just like inject your drug into somebody and then it just gets cleared by the body. You want to make sure that you're able to deliver the appropriate dosage, and that it's the easiest way to administer. So, for example, you might have, instead of being in a vial, you might want a pre-filled syringe. You want to make sure that the process is sterile, that none of the parts that you're using to store your drug are going to contaminate your drug, because that would be bad. And then you want to minimize stress conditions. So this is still a protein. They're pretty rugged antibodies, but they're still a protein. They might be subjected, have issues with mechanical, environmental stress, that sort of thing, like shaking, agitation, that sort of thing. So here are all the things that need to be considered when you're getting to that drug product process. And so here's a vial. I thought this was a cool picture. So you have your antibody. API stands for active pharmaceutical ingredient. We need to consider, which I'll go into the next slide, we need to consider what buffer we put it in, what sorts of additional salts or surfactants, stabilizers, stuff like that. We need to consider, like I said already, the delivery method. Is it going to go into a syringe or a vial? And then given the stability of the molecule, sometimes it's better to actually do like a freeze drying, lyophilization on it, and store it as a powder as opposed to keeping it as a liquid. So all these things have to be figured out. So just to give you a quick snapshot of a couple of these aspects. So formulation development happens pretty early in the development of our product. And so what we do is we take our molecule and we look at a variety of things like the pH, the type of buffer, any excipients like different salts or different amino acids, sugars, stuff like that. And we screen those at a couple different concentrations. And because we can't wait, like I mentioned, we want this stuff to be stable for at least a couple of years. We can't wait 2 years. That would just be ridiculous. So we do these accelerated studies. So we'll, instead of storing at, in the fridge, we'll store at room temperature or 40 degrees for a short period of time. We'll also look at, whether it can withstand agitation and freeze thaw. And these typically go up to a month. And so here's an example of some data, right? So high molecular weight or aggregates are immunogenic typically. So we want to keep those low. So if we look at this graph, right? This green guy, concentration 2 buffer A, not good. Over time, we're generating more aggregates. So we would rule that guy out. Whereas these concentration 1 buffer A and B, they look pretty good. So you might want to do some more work around those. So there's lots of different things. That's just telling you aggregation, which actually, if it's really bad, you can start seeing it because you get bigger and bigger aggregates and you can see it by eye. But there's lots of other things, like you might get chemical degradation, like fragmentation or changes in your amino acid functional groups, that sort of thing. So then you once you've picked your top couple of formulations, you then repeat the stability and do it it's still accelerated, but you do it at a little bit more moderate of conditions like room temperature and in the fridge. And that takes about 2 to 3 months. And then eventually you say, okay, this is my buffer. And then you do still like a development study, but you do more like a 6-month or a 12-month study on that material. And then the last piece is how do you get the drug that's in your best buffer into the vials. So there are these automated fill lines. And those are programmed to fill the vials of the drug, but there's lots of components and just your flow rates and stuff like that could impact the stability of your protein. So you have to test this out before you're ready to do your whole like $5 or $10 million batch. So when you're doing preclinical, generally you do this by hand. Once you get the clinical scale, you're doing these like single head filling lines. So it's just 1 vial at a time. And then eventually you get to these commercial lines where it's like eight heads that you're using at the same time. So that's kind of my high level overview of what we do in the biopharmaceutical development space for process development and manufacturing. And what I would say is that our underlying mission is, as drug developers, is to help the patients, but also there is quite a bit of cool science mixed in. Many diseases, like the disease that we're working on right now, don't have any treatments available. So you really feel like a responsibility to these patients to try to get them something that can help them. And there's tons of tools at our disposal to produce these molecules, but innovation will continue to be important in this area. And I think that's what keeps scientists like myself engaged, because the space is constantly moving. And the key question that I always ask is, would I give this drug to my kid or my parent, right? Because if I wouldn't do that, then it's not worth developing anymore. So that's always in the back of my head. All right, so thank you for your attention and your time. And I apologize again. I wish I could have been on site to give you this talk today, but here we are. So hopefully this is good enough. So thank you.
All right, well, thank you so much, Dr. Wollacott. And do we have questions for her? Either just unmute yourself or raise your hand.
%100% clear, I see.
Clarity, yes. Well, then I have a question since, how do you measure the percent HMW?
Yeah.
So I saw the vials that looked like turbidity of some sort. Is that right?
Yep. So we can get an early read on, there's a couple ways. So we can get kind of like an early read on the high molecular weight or aggregates using size exclusion HPLC. So we just see a population that's bigger, and that was what was used in that graph. But also, to your point, if it gets really bad, you can start seeing turbidity. So we a lot for these screening type experiments, we'll use the absorbance at 340 nanometers, because we have a clear solution, so nothing should be absorbing there. And then there's also more sophisticated tools where you can actually size those particulates and stuff like that. But we don't need that until later on in development.
Do you do any of that then, the particular sizing?
Yep. We do that, especially with the molecule. I didn't really tell you, but this molecule that we're making is a 900 kilodalton protein. So it's a huge protein. And like I showed you, it's part of its job is to stick to itself. So aggregation is an issue. And so we did for this molecule, get an early read on the sizing. And actually the regulatory agencies have limits for, you can have this number of particles of this in this size range, you can have this number of particles in this size range and stuff. So these are things you have to do before you get in, you release the drug to go into people. Yeah.
Okay. All right. Dr. Parker.
Hi, Rachel. Thanks so much for presenting on this. I think one of the things that's really important for the students to gather from your talk was how much science, good engineering there is in the manufacturing side of anything, really. So thank you for presenting that. I had a question in terms of drug discovery. So I've had a little familiarity with the CGMP and validation and all of that. Where do you start your validation package? So I'm assuming you're in discovery right now.
So we are we're getting ready to do our first manufacturing batches for phase 1.
All right.
Yeah.
So I'm assuming like your DOE information, all that is starting to become part of your validation package. You pretty much start that, I'm assuming, pretty early in your process.
Yeah. So what happened? Part of this is so there's a phase appropriate validation, right? And that happens with your process and also with the analytical methods that you're using. So there's a lot more forgiveness when you're early phase of clinical trials, because if you were to invest money and time, money-wise and time-wise in that, all of that experimenting, it would take a long time and a lot of money. And when you're first looking at safety in a small population, it's not required. So we have an idea of, okay, so our target pH for this process is pH 7.5. What happens if we were to operate at if something got messed up and we were at 7 instead of 7.5? So the more data that we have, with our process around those variable ranges, the better, but it's not really required until you get to that phase 3, like the later phase clinical trials, when you're getting ready to go into commercialization.
Great. And then the other question I had too then was, especially as you go to, I'll call it compounding, I think you called it filling, as you head in that direction, are a lot of that development. So well, I guess my question is, even when you're in the discovery phase, you're using things, materials and chemistries associated with just that manufacture. Are you bound by the USP in terms of what ingredients you can use, or is it pretty wide open? I'm assuming all the excipients have to be USP. But
they do.
But what about in the manufacture stage at this point, when you're kind of doing discovery? Do all of those have to come from USP as well?
No, it's a good question. So any preclinical work does not have to use, or even toxicology work has to use material that was manufactured in a clean room with, those fancy grade chemicals, the USP grade chemicals, or multi-compendial. That only has to happen when you get into the actual clean room manufacturing environment where you're making material to go into humans. That said, to better understand our process and de-risk the whole scaling up of our process, we do try to use the same chemicals very early on that we know we pretty much know we're going to use in the actual GMP manufacturing of the drug, because that way you just remove that variable. Because what if I've been using, one vendor that was an ACS grade early on, and it had some impurity that actually was helpful, right? And then we have a more pure, it happens.
Oh yeah.
So we try to, even though they're a lot more expensive, we try to use those USP grade, multi-compendial grade chemicals as early as possible. The research people don't really get that. So they probably aren't using it, but anything that from process development on, we try to do that when possible.
Right. Thanks again for presenting this. This was great.
Sure. Thank you.
Rachel, this is Jason. Thanks again to follow up on Dr. Parker. Great talk. I want to comment thanks for showing the value of DOE and the use of DOE to our students, because I think they sometimes don't recognize the value of statistical analysis in making decisions. One question I do have, though, is from a process perspective, as you scale up, how much analytics are done on the product, right, to make sure it falls within the specifications, right? So, we do analyticals in the research lab, but how much is done on the scale up line to validate?
Yeah, it's also a good question. So whenever I start a new program, I always think the analytical development should start first, because when you're making your drug, whether it be, the cell culture or the purification, if you don't have methods in place to understand where your impurities are, or how much protein you're making, or that sort of thing, it's not really helpful. So it's actually, analytical becomes the bottleneck for most of the process development work. So it's actually key. And, thank you for bringing that up, because I did kind of gloss over analytic, because that's a whole nother, again, that's like a whole week seminar. But yeah, so very important. And I would say as soon as process development starts, we're starting to do pretty hardcore analytics. The only ones that I would say that we we kind of wait until we're most of the way through the process development is some of the really like fine-tuned assays, like looking at charged variants. Like we use isoelectric focusing methods to look at charged variants and those sorts of things. Those are going to be dependent on your process. So we don't really do that until a little bit kind of later in the maybe second half, I would call it, of the process development timeline.
Thank you very much.
Mm-hmm.
All right, do we have any other questions for our speaker today? All right, well, I will thank our speaker for coming out, well, not out here, but coming to us virtually and talking about this whole other area. I think this is really good for us to all hear about it. We kind of get in our academia world and we forget about the whole other world out there. So this is great to hear from you.
Yeah, all and right. I always love mentoring students and stuff. So I gave Dr. Stone she had it anyway, but I gave her my professional contact information. So, please reach out if you need any advice or have any questions about career paths or opportunities. If you want to move to Boston, I could help with that, stuff like that. So definitely happy to help.
All right. Well, thank you again. And I'll see you later. Bye-bye.
Sounds good. Thank you.